rapid test for food. Today we are going to do 4 types of testing of sulfonamides, quinolones, beta-lactams and tetracyclines. The sample preparation will be the same. It will be in one step, and afterwards we will do the testing separately. So basically all these 4 types of tests, the kit, the content is similar. You have in each box 3 bottles of YRSP universal sample extraction solution, and you have 6 tubes. Each tube you have inside 8 red microwells and 8 test strips. We are going to do testing on chicken samples. Now we are going to do sample preparation. We will weigh 2 grams of homogeneous chicken sample and put it into a 50 milliliter centrifuge tube. Add 8 milliliters of our YRSP solution. In our case here it's twice 4 milliliters. Vortex for 1 minute at 3000 RPM. Finished with vortex. Now next step will be centrifuge. Centrifuge for 3 minutes at 4000 RPM. Now here we can see the supernatant. Supernatant will be tested on our 4 separate tests. So now we're going to do the testing. We're going to do 4 tests simultaneously using 1 sample preparation we've done before. First of all, we take out the required number of red microwells. Now we're going to take out 200 microliters of the sample. Be careful and pay attention to take only the supernatant. Now we will mix it well for 5 to 10 times. First incubation will be 3 minutes. Alright, first incubation has finished. Now we are placing the test strips of our 4 tests and start the second incubation of 4 minutes. Alright, second incubation has finished. Now we take out the test strips, remove the sample pad. So first thing we're going to do is observe by eye the results. As we can see, the results of, for example, tetracycline, it is more like this one, the left negative one. It's more similar to that. And the result of fluoroquinolones, the result of quinolones, it is more like this one on the center. Alright, so after observing by eye, we are going to use our ceph reader. We turn on our ceph reader, press go, press again go, click the green button, single test detection. Place our test strip and press test. Our result here, result number 1 is negative. Same with the second one, result number 2 is negative. Results 3 and 4 are negative as well. Negative in terms of our levels of detection. Negative in terms of beta-lactams, tetracyclines, quinolones, and sulfonamides in food. In our chicken sample we have checked.